What happens to the food you eat? Have you ever watched a pizza commercial on television and heard your stomach growl as the actor pulls up a warm slice with stringy cheese trailing behind? When you feel hungry, whether in response to your body's actual need for nutrients or just over thoughts of a tasty slice of pizza, hormonal signals begin to prepare the digestive system for action. How does this work when your body truly needs nutrients? A decrease in nutrient levels in your blood sends a signal to the hunger center in your brain's hypothalamus. The hypothalamus responds by triggering the release of digestive juices into the stomach. A feeling of hunger then motivates you to find food. Pause. Why do humans often eat when their bodies are not really in need of nutrients? Areas of the brain, in addition to the hypothalamus, are involved in controlling eating. Signals from sensory organs about the smell, taste, and sight of food may trigger the perception of hunger. Memories and attitudes that you have developed about food also affect your eating behavior. Regardless of how your feelings of hunger originated, the initial responses to hunger stimulate the secretion of hormones such as gastrin. Those hormones, in turn, stimulate further secretion of digestive juices in the stomach. Thus, a feedback system alerts your body that it needs or wants food. You respond by changing your behavior to locate the desired food. Pause. Humans and most other animals, sponges and parasitic worms are some of the exceptions, must bring food inside their bodies to provide proper conditions for digestion. Figure E7.8 shows the components of the human digestive system. The first steps are familiar. Obtain food, chew it, and then swallow it. Okay, figure 7.8, the digestive system of the human body. Chewing performs an important digestive function. As you chew, the surface area of your food increases greatly. Increased surface area means that the chemical reactions involved in digestion can occur more quickly. In addition, chewing moistens the food with saliva. Salivary glands located under your tongue secrete saliva. Pause. The mechanical breakdown of food is not enough. No matter how finely you chew bits of steak, the protein remains intact. If your body is to use the proteins, it must break them down into subunit parts, amino acids. Similarly, chewed bread still has its carbohydrates intact, mainly in the form of long starch molecules. Your body must break starch down into sugars to use it. Starch is a molecule manufactured in plants. As shown in figure E7.9, a starch molecule consists of many sugar molecules bonded together in a branching pattern. E7.9. How are large molecules broken down into small ones? The chemical action of special proteins known as enzymes helps make this happen. Enzymes catalyze or speed up specific molecular reactions that otherwise would take place very slowly. Enzymes act on molecules called substrates. Substrates bind to specific places on enzymes. Carbohydrate and protein molecules are substrates of many enzyme-catalyzed reactions, particularly those that occur during digestion. The reaction catalyzed by an enzyme changes the enzyme substrate into a different molecule. Enzymes that help accomplish the breakdown of food are present in your mouth, your stomach, and your small intestines. Do you remember the discussion earlier about saliva? Saliva contains the enzyme amylase. Amylase breaks down starch into maltose molecules. Maltose consists of two glucose molecules joined together. In your stomach, the enzyme pepsin binds to protein molecules. Combined with the action of other digestive enzymes in the small intestines, Pepsin breaks down proteins into amino acids, as illustrated in figure 7.8b on the page before. In general, digestive enzymes break down complex molecules into their simple components. Figure E7.10 lists some examples of digestive enzymes, the reactions they carry out, and the pH conditions under which they function. Types of enzymes: amylase, proteases, lipases, their general reaction, and optimal pH. After you swallow, it takes less than 10 seconds for the chewed and moistened food to pass through the esophagus into the stomach. Smooth muscles encircle the digestive tract. These muscles contract in a coordinated fashion, called peristalsis, to move food through the digestive tract. In the stomach, food churns and mixes with digestive fluids for 3 or 4 hours. The stomach stretches during this process and makes you feel full. This sensation reduces your motivation to continue eating, unless the food tastes so good that you ignore these signals and continue eating anyway. This is another example of feedback which you studied in Chapter 5. From the stomach, partially digested food passes into the small intestine. Final digestion of the complex food molecules, breaking down into their simple components, occurs here. The cells that line the small intestine contribute fluids that contain digestive enzymes. Other organs, including the pancreas and liver, also deliver digestive enzymes to the small intestine. The pancreas is an organ in the abdomen that produces 1.4 liters of fluid per day. The fluid contains enzymes that contribute to the final digestion of the remaining macromolecules in the small intestine. The liver produces 0.8 to 1.0 liter of bile per day. Bile is stored in the gallbladder and released into the small intestine after a meal. Bile contains bile salts, which act in a manner similar to detergents. Bile breaks fat into tiny droplets, increasing the surface area of the fats so that the enzymes can work on them easily. In addition to being the site of digestion, the small intestine is the organ where nearly all absorption takes place. The digested nutrients consist of simple sugar, amino acid, and fatty acid molecules. These building block molecules, obtained through digestion, are small enough to pass through the small intestine cell membranes and into the bloodstream. Liver cells monitor nutrient levels in the blood and adjust them as necessary. Substances present in excess amounts are removed and stored. Substances that are lacking are increased. The liver accomplishes that by releasing stored forms or by stimulating synthesis processes. For example, when you exercise heavily and glucose levels drop in your bloodstream, sensors trigger the liver to release glucose that it had stored as glycogen. The glycogen is quickly broken down into glucose to replenish the exhausted supply. Liver cells also remove and correct potentially toxic substances, such as alcohol and other drugs, from the blood. Undigested remains of food cannot pass through the wall of the small intestine. Instead, this part of the food enters the large intestine. The large intestine absorbs water and returns it to the blood. The body then eliminates the compacted solid waste.